Welcome everyone to the Ecological Continuity Trust's 34th webinar in our series. Today we have two speakers, Dion and Lyndall, who are going to be talking to us about the Rothamsted Insect Survey and the BIOSCAN Insect Biodiversity Project. Before we start that, just a little bit of housekeeping. If I can ask you to turn your cameras off and keep your microphones muted during the seminar. If you have any questions, as you think of them, please do pop them in the chat. We'll be going through the chat to look at questions at the end. You'll find the chat, if you just look in the bottom right-hand corner of your screen, there's a little icon for chat there. So do please put any questions you have. So our first speaker will be Dion. Dion's a molecular entomologist with expertise in integrated pest management and agroecology. And he's particularly interested in detecting plant pathogens and their insect vectors in a variety of agricultural systems using molecular tools. We'll also be hearing from Lyndall. Lyndall has a background in aquatic entomology, taxonomy, and invertebrate biomonitoring, and she joined the BIOSCAN team at Sanger in 2021 as a samples and project manager to develop high throughput pipelines for the collection of DNA sequencing from UK flying insects. So over to you, Dion.
Lovely. Yeah. So yeah, thank you very much, Kylie, for the introduction. I've been just a little bit more. Yeah, I was a PhD student for the Waitrose CTP starting in late 2018. I was based at the Rothamsted Insect Survey in Harpenden. I then went on to do a postdoc within the Insect Survey as well, and I'm still at Rothamsted, and as Kylie mentioned, I'm sort of more of a molecular entomologist, integrated pest management specialist now. So I'm outside of the team, but I still have a lot of interactions with the Insect Survey as well. So I'll just go a bit of an overview of what the Rothamsted Insect Survey is, because I think the audience is quite diverse. And yeah, as Kylie said, please put any questions in the chat. But predominantly I'll do a bit of an overview of the suction trap network and the light trap network that Rothamsted Insect Survey runs and manages. And then I'm just going to go on to just a few sort of research points from some of the current and past PhD students at Rothamsted, and just really just to demonstrate some of the work that, you know, without the Insect Survey, it wouldn't be possible. So the Insect Survey, as I mentioned, they constitute essentially two distinct networks. So the first one is a light trap network that was established in roughly about the 1930s. There's currently now about 65 light traps within the UK. And these light traps are focused predominantly on catching nocturnal insects, but mainly micro and macro moths. And the suction trap network, established 1964. There was 16 traps. I think we're down to 15 traps now, but we've just got another one erected in Harper Adams site, which is fantastic. And that really focuses on aerial flying insects, or what some people sort of term aerial plankton, because the suction of the suction traps really isn't too strong, so it's more sort of catching the smaller insects, and this is what we're very interested in. So this is catching aphids, which are quite a significant agricultural and horticultural pest. And both of these networks, they represent the most comprehensive, standardized long-term data on insects in the world, and it's a really incredible and valuable resource that we have. So the suction trap network, as I said, there's about 15 throughout the UK. So the UK, Scotland, and we've got a few in Ireland as well. They're 12.2 meters tall. This sort of height was optimized in the early 60s by a scientist called Taylor. He sort of measured various different heights and essentially deducted that 12.2 meters was roughly about the sort of optimum height to catch the greatest number of aphids, but also provide a really good representative of sort of aerial flying insects within a sort of 80-kilometer, 50-mile radius. So here's the current distribution we have of the suction traps. These suction traps operate 24 hours a day, 365 days a year. They're sampled on a daily basis in spring, summer and autumn, and they move to a weekly sampling in winter. And the idea here is predominantly, as I mentioned, looking at aphids. And just highlighted, this is essentially a bulletin. I pulled this off. This is from quite a few years ago, but it's just to represent what the insect survey is trying to do and does really well. And these are a list of the sort of main horticultural aphid pests. On the left hand side, there's 21, and then along the top, these are just the different sites, and this is individuals core of those species in a given week. So I said there's only 21 included in that bulletin. So, but there is actually over and around 625 aphids we have in the UK, 400 of which are caught in the Rothamsted Insect Survey traps regularly. And the reason why only 21 are reported, because these are the main horticultural ones. So a lot of the other aphids are actually regarded as quite beneficial. They may be can be a bit of a nuisance on your roses and things you're growing in the garden, but they actually do provide such a valuable resource for other insects and also the environment too. As you can imagine, the taxonomists, it's a really big job, and it's very difficult to speciate aphids. So it takes between 3 to 4 years of training. This is all done manually at the moment, just sort of illustrated in the bottom right picture there. So these taxonomists have trained for several years to actually get to the level where they can speciate aphids by eye. It's pretty fantastic work. But we're also looking at expanding that now. So very familiar now, I'm sure everyone is, with artificial intelligence. You know, it's a bit of a mixed bag, but we're sort of testing the water to see if we can actually differentiate between aphids and other insects, because a lot of time is actually spent on sorting out aphids from other insects, and it's very laborious, and this can create some bottlenecks, especially during sort of very high seasons when we're getting high pressure periods of time when we're getting a lot of aphids coming through. So this is just a bit of work from the University of Lincoln and the Insect Survey, and it's just sort of highlighting here. We've got the sort of bounding boxes here, just sort of identifying aphids. Some it's not identifying as well, so it's a work in progress. But this is using a YOLOV 5 system, and in terms of trying to ID. So that's some work we're sort of looking at, trying to reduce some of those bottlenecks. There's also another institute, Julius Kahn Institute in Germany, started a bit of a collaboration with them, and they've moved kind of a step further than that. They're actually able to differentiate or speciate aphids using this particular system, which is showing some really promising results, and that could really help aid the taxonomy, especially in terms of time for when sample numbers reach very high levels, and it's quite taxing to get everything done in time for the report. So in terms of the report, so the website here, just you've got the link at the bottom of the screen. So this is freely accessible, and what it's doing is it's highlighting. So these are the highlighted points here. So these are all the suction traps which are currently in the network, and you can actually go into the sort of archive bulletin, and you can retrieve past sort of numbers of different aphids. You can also get an aphid forecast. So this is one of the reasons why the Insect Survey is regarded as a national capability, because it can actually help forecast when aphids are likely to come into crop or sort of into the area. So it essentially provides a really nice early warning system as well. Additionally, on top of that, the aphids are all kept. So all of the aphids and other insects, they're all sorted, separated and stored in samples in the archive, and we have samples, going back to the 60s for aphids and for other insects, and this is where this resource is incredibly valuable for sort of academic work. So onto the light trap network. So as I said, this was established in the 1930s. Trap ranges vary it can be quite sporadic just because a lot of the network is volunteer run, but there's about 65 traps currently in operation throughout the UK. Again, these are sampled on and collected on a daily basis, and they are sent off to trained moth taxonomists, but it is predominantly volunteer led and run, so we don't get sort of very fast information back, but this is more of a sort of a service to create and understand the changes of nocturnal insects over time. So there's about 2,500 moths in the UK, 900 of which are macro moths, and the remaining are micro moths, which are incredibly difficult to ID by microscope. It's typically dissection is required in order to speciate the micro moths. But again, a really valuable resource. And what the sort of light traps we have here, we just have essentially a tungsten bulb. It emits a really good wavelength over all of the near visible and visible wavelength bands, very attractive to moths. We actually have this cover on the top as well, and the idea behind this cover is any moths that are flying overhead won't see and therefore won't be attracted to the light. So the idea is to really just get what moths are within a particular area or habitat, and not the ones which are flying from or between different habitats. And so we've got the bulb here. Moths are attracted. We've got essentially a fluid which is sort of I think it's tetrachloroethylene, which will kill the moths, and unfortunately they have to be killed because we can't ID them without. And a lot of moths, when they fly into a light, they'll also fly away, and this is how it's been done since the early 60s. And to maintain the sort of long-term standardization, we're keeping it the same for such purposes. We've also got a sort of sensor on the top as well, and that is just a light and dark sensor. So when it senses dark, the light comes on, and it's powered by main supply as well. So I mentioned there's 65 traps currently in operation. We've got a few in Ireland as well. What's really interesting here is we've got all these different traps are associated within different habitats, and that's what I said. That's why the top of the light trap is really important to have, to actually understand what's going on within sort of the local environment. So again, this information and data is accessible on the Insect Survey Moth Data website. You can also request data as well from the archives. So any reasonable requests are accepted, and providing the Insect Survey is acknowledged, the data can be used freely. And then we've also got a very big data warehouse as well, which we have all of the moth species recorded, and you can actually go, and if you're interested in a particular species of moths, you can request and get graphs and data from that as well to look at. And on the sort of progressing the identification front, as I said, both of the light trap network and the suction trap network is so powerful because it's long-term standardized data over time. This is some really good work from the Centre for Ecology and Hydrology, or the CEH. And they've actually put together this AMI system, and it's essentially automatic monitoring of insects. Same principle, but we have a light up here on a piece of sort of strong card. We've got a camera as well in here, and what that does is it images anything that lands, any nocturnal insects which are attracted and lands on this plate. It's a Raspberry Pi system in terms of its processing, and that information is once sort of identified, or what it believes is a moth or a caddisfly, etc., is reported back to the operator. So these are completely remote. They've got solar panels as well, so it doesn't require main supply. And these have actually been sort of reproduced and used within various different countries with a lot of success. So I'm interested to see sort of what happens with that, but it's just really just to demonstrate some of the other ways that we're monitoring for insects too. So that's the two networks. That was a sort of a whistle-stop tour, but hopefully it gives you a bit of a flavour of what the Insect Survey does and is about. And what I'm just going to go on to now for the next sort of few minutes is just some of the research that has been produced from a few sort of PhD students at the Insect Survey. This isn't an exhaustive list by no means. We've got a lot of technicians and other researchers who have used Insect Survey data, but this is just the past sort of few PhD students who have come from the Insect Survey. So we've got Hannah, and then we've got who's looking at bat, particularly common pipistrelles and soprano pipistrelles, and trying to understand the relationship between nocturnal flying insects and understand their diet via DNA metabarcoding. Elliot, as you can tell, he's a moth guy. He's doing some really interesting work on moth visualization, so how they actually visualize and interpret light and how they're attracted to it. Ishbel's another past PhD student. She did some really good work, again, on light traps and trying to optimize and understand different wavelengths and patterns and what attracts moths to sort of help improve the light trap network. And Dan, again, covered in moths, clearly a moth guy, and he's done some really good long-term trend analyses using the Insect Survey data as well. So I'll just very briefly go over some of the work that we've done, and then myself as well during 2018 and 22. So I worked on mainly this was the stuff from the suction trap network. So I was looking at the current lettuce aphid, Nasinova rubus nigri. It's a really severe pest of lettuce. There's very little to no effective control strategies, and it's now resistant to a once resistant lettuce cultivar. So this was a host plant resistance within the lettuce. There wasn't a lot of molecular data, so I assembled a couple of genomes of both the resistant and wild type. So this is resistant to the host plant resistance, and a wild type which was susceptible to the host plant resistance. And this is just the pipeline that I used to assemble the genome. It's just a hybrid assembly, essentially using two different sequencing chemistries to create a nice genome. And this is just a comparison with some other aphid species which are on the sort of the Barcode of Life and NCBI, just comparing that. They're quite good genomes, so I was really pleased with them, and that work was published last year. I also did I sort of did a bit of a deep dive into transcriptomics as well, and I was trying to really understand what this potential resistance mechanism was in the resistant biotypes of the aphid. So this one here, this is a differentially expressed gene analysis, and really it was highlighting this one particular gene was being significantly upregulated between the resistant and susceptible aphids. So I then did a analysis using the susceptible aphid, so the one which wasn't able to feed on resistant lettuce, and then 3 different cultures which were, and it was shown that the expression of this particular gene was incredibly overexpressed in comparison to the susceptible. So it sort of confirmed that this was potentially the gene involved for the resistant breaking mechanism, which I was really pleased about. So I've also done a bit of population genetics work. So this is on, again, using on the same aphid, but this was just to demonstrate how far the archive goes back. So this was 55 years. There's about just over 7,000 individuals of this aphid that I was studying, and I just did a population trend analysis. And even though it's still a really big pest, it's actually declined quite significantly. This is throughout the UK, and then split this between sort of the north, south, east and west as well. And yeah, within whichever region it's been in, it's actually still in decline, but yet it's still quite a severe pest of lettuce. Also done some population genetics analysis. This was really just, so this was going back in the archive, and I had to go through, pick out all the different aphids, and find the one that I was interested in. It took absolutely months to do this. This is just a plot just showing the amount of DNA that I got from all the different regions of this aphid, and some were, they were sort of close to 20 years old, and we're getting some really good numbers of yield. So there wasn't really a correlation between time and DNA duplication, which was quite interesting. And then, yeah, just sort of finally on the population genetics, I just did a genetic clustering, and I sort of this is illustrated here, and it's just showing two different clusters of these aphids. So this was 20 years of data, about 400 individuals, and this really just shows that there seemed to be a difference between east and west, and predominantly a lot of lettuce cultivation it goes on in the west compared to the in the east compared to the west. So that's some interesting data that I got from that. Moving on to Hannah, so she's still in her PhD at the moment. She's submitted one of her chapters, and that's been accepted, which is fantastic. And this one was just a short communication we did using the insect survey data and looking at swift boluses. So when swifts come to their nest to feed their chicks, they will regurgitate a thing called a bolus, and within that bolus is insects that they've caught in a sort of a flight path. So one of the swift ringers, he actually was sort of ringing some of the chicks, and some of the adult swifts would come in and they'd regurgitate these boluses. And what he did was actually collect a few of them, and what me and Hannah did, amongst a couple of others at the Insect Survey, looked at their sort of diet and what they were eating. So this was comparison using the Insect Survey suction trap at Broom's Barn, and these samples were collected in Stonham, which is in Suffolk, and it was really showing that there's quite a big difference between what they were catching, which was flying at the same time, sort of within and being caught in the suction traps. So it seemed like they're selectively feeding on a lot more Coleoptera or beetles. We found that they're catching quite a significant number of cabbage stem flea beetles, as well as pollen beetle as well. So it was a really nice analysis that came out of that, and a really good short communication, again using the insect survey data. And finally, Dan Blumgart, he finished his PhD in 2022, and he was obsessed with moths, still is now, still does light traps. He lives in Costa Rica now, and he started his own sort of entomological venture out there, which is fantastic. But he published this work in 22, and he showed that there's a really large decline of moths, most severe in broadleaf woodlands. So these are the 7 different habitats that he analysed, and this was over a 48-year period. And then you've got these are the different sort of feeding gills. So you've got very highly polyphagous that eats a lot of different plants. You've got lichens, conifers, and he showed that over time, this is a percentage change over time, showed that there's actually quite a significant decline in broadleaf woodlands. So really good work there. He's done quite a lot more. He interviewed for Radio 4 as well, and it's a great talk. So we're getting quite a lot of good information from using the insect survey. So that is everything. Sorry if I've gone over time a little bit. There's quite a lot of information to put in there, but hopefully that's given you a bit of a flavour of the work we've done and are doing at the insect survey. Thank you. I think it's over to you, Lyndall, isn't it? I was meant to just pass it on.
Yeah, thanks.
Brilliant.
Okay, hi everyone. So back in February 2023, I gave a webinar on BIOSCAN, and that was sort of the setup and where we were with the project. And so now I'm going to give everyone an update. So as a quick recap, BIOSCAN is a Tree of Life project at the Wellcome Sanger Institute. The Tree of Life program focuses mostly on whole genome, reference genome sequencing, and as part of the Earth Biogenome Project, BIOSCAN focuses on shorter read DNA or DNA barcodes, and we're affiliated with the International Barcode of Life. So the project sort of aims to future-proof and biobank DNA. The project scale is 1 million arthropod specimens processed throughout the project, and that's collected from 100 Malays traps across the UK. We have a partner commitment of 2 to 5 years and monthly traps, so every month throughout the year for 24 hours. We have a short catch time so that the catches can be individually plated, and it gives us a higher quality DNA extract if it's a shorter time. So the partners then plate out the entire catch, they record key information, and they ship the samples to us. So in the talk in 2023, we had 34 traps, and so we've had a pretty good scale up of our network, and we currently have 102 active traps. And on the right-hand side is a little word cloud of our partners, and the larger the text, the more traps they have across the country with us. So that's National Trust, UKCEH, Natural England, and Rothamsted Research are our bigger partners. So here is our pipeline in a nutshell. We do a non-destructive DNA extraction so that the specimen is still available as a voucher. So right on the left-hand side, we have our Malays catch, which is then plated out by partner and sent to us. When it arrives to us, we image each specimen and we create a bold record with all the metadata. And then those specimens, after the DNA is extracted, they become vouchers and are either returned to the partners if that's what they want, or deposited in museums for sort of future research. And the DNA that we use for our sequencing is only 0.1 microliters out of 100 microliters. So there's still a lot left over for the biobank. And at the moment, we are sequencing about 20,000 specimens per month on PacBio. And then from that point, we do all of our sort of downstream bioinformatics using mBrave, so it separates all the sequences out to individual specimens. And then we run our own QC on the quality of the sequences. So they either get passed, put on hold, or they fail. So on hold is when we have more than one, sequences from more than one species in the same sample, and so we can't tell which is which, so they're on hold for review. And from there, we submit our sequences into BOLD for species ID. We compare to what we put it through the BOLD ID engine and the classifier tool to get an identification. And then they also get a bin assignment. So as a quick recap on what a bin is, it stands for barcode index number. It's a species proxy because the reference database doesn't have a name for everything or a sequence for everything. So essentially what it does is it just clusters similar DNA sequences together, and this often reflects a species, but it can also distinguish between populations of the same species, depending on the taxa. So once we have an ID for all of our past sequences, this updates the BOLD record, so all of that metadata and downstream results are together. And then at this point, we what we're trying to do is try this approach where we actually enhance our data as well as enhance the databases for the UK and other projects that might be using reference libraries and databases. So if we zoom in on this little icon, our aim is this sort of enhancement and integration and then adding value to BioScan data. So we're working quite closely with the UK Barcode of Life at the Natural History Museum, Natural England in particular with mobilizing DNA data, and then NBN Atlas, which is where we would like to submit our records. So essentially, we have our barcoding that we've uploaded to the BOLD database, and this can be parsed sort of into two. So we've got records that match the UKSI, which is the UK species inventory, and anything that sort of matches is probably quite confident. So those can go to the NBN Atlas. And then things that don't, so they either don't have any identification, there's a conflict, or they don't occur in the UK, they are sort of flagged. And then from this point, we can get an expert review in. So that's UK Barcode of Life or expert collaboration with taxonomists or recording schemes. And then once they've had a look at these records, they do some reviews and verifications, then that enhances the database, but there's also this sort of informing and comparing with UKSI. So species that are actually new to Britain, then we can update the inventory, and then those records can get added to the NBN Atlas. And then this, of course, contributes to GBIF, and hopefully then has more of an impact on species diversity knowledge, and using platforms that already exist, and our goal of having this impact on biomonitoring and conservation. So one of the ways that we are, one of our approaches for this expert review and collaboration is hosting taxonomy workshops. So we've got our first one that we're trialing in September this year, and we provide those that will be attending with comprehensive data reports depending on the group that they are familiar with or expert in. So that's our report cards, which I'll show you in a minute, the images, as well as some other bits of information, like you can see on the right-hand side with the UK Barcode of Life website, that it shows you a bin number, and then it shows you a bunch of names. So that this happens where there's this sort of discordance between the species names and taxonomic names, and then how the DNA is clustering. And this is really useful because it can really focus those efforts for experts to have a look at these and sort of fix it and improve the database. So what progress have we made so far? In the overview, taxonomic breakdown, we have, not surprising for Malaise traps, 74% Diptera, 13% of its Menoptera, and 5% Hemiptera. So far, we have 230,000 specimens submitted to Sanger. There are 60,000 in the pipeline. We're always about 3 months behind with our sequencing, so it's, we do 20,000 a month, and we have 170,000 sequenced. 82% of those have a high confidence pass, and that is where there's one very clear sequence that has come from the specimen, and a 5% on hold that we still are working on the best way to review. And then at the moment, 13% failure rate, but this does improve as time goes by. So, and then this from our report card is just sort of to see how, especially shows our ramp up from our pilot and sort of our first year getting going to 2023 and 2024 when we really ramped up the project. And this is really rough and ready comparison, just looking at using the UKCH land cover maps, how much do our sites represent the UK habitat coverage? And so it's quite similar, but we're probably missing a couple of sites with Mountain Heath and Bog and Arable. So if anyone watching this would still like to be a BIOSCAN partner, we are, if you're in these habitats, by all means get hold of us and we'll send you a trap. In terms of bin frequency distribution, so we have found 9,000 bins so far, so 9,000 species proxy. Over 3,000 of those have only been observed once. This may be a sampling artifact because we have done a very quick ramp up. So we do expect this number to drop. But what's also interesting is that we have currently 300 bins that have been observed more than 50 times, which is great for population genetics and other sort of community network analyses. And on the right-hand side, we have our top 3 bin species that have been seen over 2,000 times so far in our data set. And what I find really interesting about this is that only one of them actually has a species name. The others don't have a reference on the database, and so we just get spat. So back to our pipeline, we're right at the end on our biodiversity report card, and I have a short video clip to showcase the report card. I showed a clip in my last talk, but this sort of has upgraded somewhat since then. So this is what the report card looks like. You're familiar with this little sunburst plot. It's interactive. You can click through, and it's also linked to this bar plot that I've shown you. So you can click through whichever taxon levels you're interested in. And we also provide a table that is fully downloadable to get your data. we show and display our QC result with a description of why it's passed or failed or on hold, and also to the UKSI. You got synonyms and recommended names, and if it's blank, it's a failed sample, unfortunately. And if we click on the unique specimen ID, you can also have a look at your record on bold with the images and the other data. And we also pull some information from Pantheon to hopefully enrich the data just a little bit with some ecological data and conservation status, broad biotope, and also this specific assemblages and some links to the Pantheon information. So what now? Well, we have a postdoc who's joined our team, who's going to do some really awesome analysis on the spatial temporal aspect of our data. And she'll be looking at shifts in species distributions and modeling and community compositions and networks and in response to the environment, which will be very exciting. And there's also this potential to explore a smaller subset of the DNA biobanks, looking at population genetics and where there is species hyperdiversity. And another really interesting thing that has come from our data is that with just sequencing the CO1, there's a lot of species interactions that we're seeing in secondary DNA sequences that we've picked up per sample. So one in particular was from Bifurface, which is a Drosophila where there's evidence of this nematode that is known to infect mushroom-feeding fruit flies. And then we have multiple other examples of arthropods that have evidence of nematodes or quite specific nematodes and rotifers, as well as plenty of arthropods with evidence of Wolbachia. So there's a lot to unpack there, which is very exciting for our future analyses. And as a sort of ending slide, we really want to maximize the impact and the use of our data. We're a very small team, so if you would like to collaborate with us, please do get in touch. Our email's at the bottom there. Broad areas where we're quite interested in working on is enhancing conservation monitoring, evidence-informed policy, establishing baselines for various research, and guides to adaptive land management. So with that, thank you so much for listening, and thank you to all the people on my acknowledgement slide who have contributed to the project and made it possible.